Hello to everybody. Here, first of all, I'm going to interview Judit Cubedo, who's one of the key persons within the Proteomics platform here at the Cardiovascular Research Center in Barcelona, and who has been leading the performance of this podcast in Proteomics. Hello, Judit Cubedo. Thank you so much for being here today.
Hello, Gemma. It's a pleasure.
So my first question regarding proteomics would be, what has proteomics offered to the research community overall?
In general, the introduction of omic technologies has induced a profound change in the biomedical research, and specifically the genomic approaches allow the identification of all the genes, the encoding genes, but the proteomic approaches will allow us the identification of the specific protein networks that are actively expressed in a specific situation. Indeed, proteomic technologies allow us not only the identification of several proteins in a single experiment, but also the analysis of protein interactions and the identification of protein post-translational modifications and protein variants. And this is important because this will allow us to identify novel signaling pathways, also identifying new potential biomarkers and novel potential therapeutic targets.
Great. So, with this in mind, do you think that we can do proteomics of everything?
Of course, we can analyze cell extracts, tissue extracts, and also more complex samples such as serum or plasma or urine. And these samples are considered complex because they have a wide range of protein abundance, and there are a few proteins that represent nearly 90% of the protein content in these samples. And this means that when performing proteomic technologies with these samples, we must apply a previous step, a prefractionation step to the sample in order to reduce their complexity.
Great. So, as you have just told, proteomics seems a really complex platform, and which not only you will talk about in the podcast, a two-dimension polycrystalline amyl gel electrophoresis, mass spectrometry, databases analysis. So, do you think that any lab can have it? Do you think anybody can do proteomics?
Well, the establishment of a proteomic platform is complex because, as you have said, you need sophisticated instrumentation, you need advanced softwares, and you need well-trained people. Specifically for the traditional approach, which are bidimensional electrophoresis, you need skilled operators in order to obtain reproducible results, because these technologies cannot be automated. Mm-hmm. On the other hand, the mass spectrometry technologies require a high-level computational and statistical skills, because you generate a huge amount of data and you need to efficiently analyze this data.
Okay. So, when said that, you have given some insights about what we're going to see in the podcast, its complexity, what can it deliver. So, what are the challenges remaining within the platform of proteomics within the future?
Well, I think the major challenge would be the integration of all this data that we obtain in general with all the omic technologies to know more and specifically of each organism and each disease state, what is known as systems biology. This is a multidisciplinary approach that integrates information of omic technologies with bioinformatics and statistical analysis tools. And specifically for proteomic, technologies have rapidly evolved, and nowadays when you perform a proteomic study, you obtain a huge amount of data, and you need tools to efficiently analyze and process this data. So we need that bioinformatic and statistical analysis tools will evolve in the same rapid way in order to be able to respond to this huge amount of data that we generate with proteomic technologies. And specifically when performing biomedical proteomic studies, you need to know the specific scientific question you are going to answer in order to be able to design the experimental approach, and this will allow us to reduce what is known as chance findings, no? Okay. That are so common in proteomics. And also, more specifically, when you want to perform a discovery study, a differential proteomic study, to identify novel biomarkers or novel potential therapeutic targets, you have to choose the proper controls in order to obtain the reliable results you are searching for.
Okay, great. Thank you so much. Hopefully the big pharma and the technological industries will really help in the development of more powerful tools. And just thank you again, Judit Cubedo, for the insights. And let's have a look at the podcast.
In this podcast, we will go through a general overview in differential proteomic studies and show a practical example of a proteomic experiment. Traditionally, genes and proteins were analyzed one by one, but with the advent of omic approaches, we are now able to analyze several genes or proteins in a single experiment. Thus, the application of genomic approaches tell us what could happen. Transcriptomic approaches give us information about what might happen, and proteomic approaches are able to tell us what is actually happening in a specific scenario. Traditionally, the most commonly used omic approach was genomics. Genomic approaches allow the identification of genes associated to a specific disease and thus the determination of potential candidates for predicting the risk of suffering a specific pathology. However, a single gene can produce several proteins due to alternative processing and post-translational modifications. Indeed, the genome is stable, but the proteome is dynamic, as it reflects the biological processes that take place in a specific pathophysiological situation. Thus, while changes at a genetic level are risk markers, modifications at a proteomic level afford additional information about the different mechanisms that lead to the development of a pathological state. During the last years, proteomics has become a basic strategy for the study of the protein profile and complex protein associations in a biological sample. By the use of proteomic approaches, we can determine modifications in protein structure, expression levels, and the presence of protein complexes and protein post-translational modifications that can be associated to a specific disease, having thus a diagnostic, prognostic, or therapeutic value. Proteomic approaches are a two-step technology. The first step is protein separation. This step can be a gel-based separation methodology in 1 or 2 dimensions, after which proteins are stained to be visualized, or a gel-free separation methodology in liquid phase, such as liquid chromatography. Both protein separation approaches have several advantages and disadvantages. In gel approaches, specifically bidimensional electrophoresis gives information about the isoelectric point, providing effective filter data for peptide protein identification. Moreover, it affords the highest resolution on protein level, allowing the easy visualization of protein post-translational modifications and isoforms. In addition, it affords quality control for sample preparation, as an inadequate treatment or protease activity are easily recognized by shifts in protein patterns. On the other hand, this approach has limited usefulness for very hydrophobic, large, basic and low abundant proteins. Moreover, it requires skilled operators and cannot be automated. Gel-free approaches afford high sensitivity, allow the identification of peptides of basic, acidic and hydrophobic proteins, and can be automated. However, with these approaches, the sample complexity is increased. There are important difficulties in the assignment of peptides and post-translational modifications, and furthermore, once proteins are digested, the information about protein degradations, isoforms, and post-translational modifications is lost. Therefore, it is important to highlight that both approaches are complementary and one cannot replace the other. The second step is protein identification by mass spectrometry. Mass spectrometers are classified based on their 3 main parts: the ionization source, the mass analyzer, and the ion detector. Among the ionization sources, the most common ones are the matrix-assisted laser desorption ionization, or MALDI, that is used for solid samples, and the electrospray ionization, ESI, that is used for samples in liquid phase. MALDI is more appropriated for high molecular weight compounds as proteins, and ESI allows the analysis of a wide range of molecules from peptides to proteins. After the ionization, precursor ions are filtered in the analyzer depending on their mass-charge relation prior to the detection. There are different types of mass analyzers, such as ion separation by time of flight, what is known as TOF, separation by electromagnetic camps generated by quadrupoles, or the separation by ion trap. Specifically, differential proteomics allows the comparison of the proteomic profile of samples from different conditions. In the discovery phase, the coordinated proteomic changes that occur in a specific situation are analyzed. After the discovery phase, differential selected proteins are characterized and validated by using complementary methodologies. In each step, the sample size requirement increases. Thus, differential proteomic studies are based in the integration of several technologies that include sample preparation, protein separation and differential protein profiling, protein identification and characterization by mass spectrometry, and bioinformatics analysis in silico to determine the potential involved molecular pathways and networks. This study design is very useful for the discovery of novel disease biomarkers, the development of novel therapeutic targets, and the identification of new disease-related pathways. The classical differential proteomic approach includes protein separation in 2 dimensions that is known as bidimensional electrophoresis. In the first dimension, or isoelectrofocusing, proteins are separated by their isoelectric point in dry strips with an immobilized pH gradient, and in the second dimension, proteins within the strip are separated by their molecular mass in a traditional SDS page electrophoresis. Proteins in the gel are then visualized by specific staining, and protein spots of interest are excised, enzymatically digested, usually with trypsin, and identified by mass spectrometry. Afterwards, in this podcast, we will show a practical example of the steps of a bidimensional electrophoresis experiment. This is an example of a MALDI-TOF. Samples are mixed with an organic matrix and then loaded on a plate that is placed in the MALDI. Then the laser of the ionization source impacts on the sample, accelerates the peptides, and the peptides fly through the time of flight tube. In this system, the time of flight is proportional to the mass-charge ratio of the peptide. This means that the smaller molecules fly faster than the larger ones. It is also possible to work on the reflector mode in a way that ions are reflected towards the detector, increasing the accuracy of the measurement. In this system, proteins are identified based on their mass fingerprint after digestion with specific enzymes, such as trypsin, and comparison with databases. Identification can be improved by peptide fragmentation using tandem mass spectrometry. In differential proteomic studies, the proteomic profile of different conditions is compared by specialized computer programs in which every spot in the gel is assigned as a relative value. Then the spot intensity in each condition is compared and a statistical analysis is performed to detect proteins or protein isoforms that are increased, decreased, or change their distribution profile in a specific condition. This type of analysis allows not only the identification of proteins that are up or down-regulated in a specific pathophysiological situation, but also the identification of changes at a post-translational level. This is crucial as the relevance of a protein in a specific disease is not only through its total levels, but also through its structure and the presence of post-translational modifications. Once identified, differential proteins can be analyzed with specialized bioinformatic tools to identify the functional and molecular networks in which differential proteins are involved, in order to detect the significantly modified signaling pathways in a specific condition. The first step is an overnight rehydration of the dry strips of immobilized pH gradient prior to the isoelectrofocusing. The strips can be rehydrated directly with the sample in an urea-containing buffer, as we are doing in this case. This step can be passive or active if it is done by the application of a constant voltage through an overnight step. Once the strip is rehydrated, it is placed in the tray to run the isoelectrofocusing. The isoelectrofocusing program consists of several steps in which the applied voltage progressively increases. The number of steps and the voltage depend on the type of sample. For specific samples, strips are rehydrated with a buffer without the sample, and the sample is loaded into specific isoelectrofocusing caps. This is known as cap loading. This procedure can be done through the anode or through the cathode, depending on the nature of the sample. For example, cell membrane protein extras must be loaded by anodic cap loading. Afterward, strips are covered with oil to place the tray in the isoelectrofocusing chamber. Then we place the tray within the isoelectrofocusing cell. We select the specific lane where we have placed the strip. And we choose the length and the pH gradient of the strip that we are running. In this case, this is a 70 centimeter strip with a pH gradient 4 to 7. Then we have to choose if we want a rehydration or not. In this case, as we have previously rehydrated the strip, it is not necessary. We close the lid and we run the protocol. Once the isoelectrofocusing has ended, strips are equilibrated in a glycerol containing buffer, supplemented with a reducing agent in a first step and with an alkylation buffer in a second step. Afterwards, the strips are placed on the top of SDS stage gels and sealed with agarose. The agarose must be hot to be liquid, and we have to wait until the agarose is solid. Then gels are placed on the electrophoresis tray. Then gels are placed within the chamber. And the chamber is filled with running buffer. We connect the electrodes. And the program is set on. The running conditions will depend on the system used and the number of gels. Once the electrophoresis has ended, proteins in gel are fixed and stable. Gels are taken off the glasses. If the strips are shorter than the gels, the remaining part of the gel can be taken off. And the gel is placed on fixation solution. Staining can be done with chromacy blue, silver, which is more sensitive, or fluorescent stainings, which are as sensitive as silver but do not interfere with mass spectrometry analysis. When fluorescent staining is used, gels must be protected from light. If the dye technology is used, samples are directly stained with fluorescent dyes prior to isoelectro focusing, and gels don't have to be stained for protein visualization. When gels are stained, we obtain the images. This gel has been stained with a fluorescent staining, and we will acquire the image with a specialized scanner. We place the gel in a glass. And then in the computer, we select the specific area where we have placed the gel, and we start the scan. The scanning process can last for 5 or 10 minutes. Once the scan has ended, we obtain an image where every protein or protein modified form is represented by a specific protein step. Then we perform differential analysis of all the images from the same experiment with a specialized software to identify protein spots with a differential distribution profile between samples. Once we have scanned the gel, we put it into a spot picker where we will excise the protein spots of interest. Protein spots of interest that are selected will be excised from the gel automatically with a spot picker. These selected spots will be identified by mass spectrometry. Once spots of interest are obtained, they are washed, dehydrated, and dried before an overnight trypsin digestion. Then peptides are washed and concentrated with a tip filter. Samples are applied to a pre-spotted anchor chip plate, and once dried, an organic matrix is applied to the sample and allowed to crystallize. Afterwards, the plate is introduced in the MALDI and the program is set to acquire the spectra considering a specific signal-to-noise ratio, applying statistical calibration and eliminating background peaks. Identification is carried out by peptide mass fingerprinting, and identified proteins are confirmed by peptide fragmentation working on the reflectron mode. Spectra data are then compared to information included in databases.